Okay, next item on the program is a talk by Professor Florian Krammer. He's a professor of vaccinology in microbiology department of IISc School of Medicine. He's been pretty much leading the field of developing serological assays, understanding the antibody responses since the SARS-CoV-2 emerged. Other than that, his major focus has been trying to develop the universal influenza vaccine strategies. Florian, may I request you to turn on your audio and video?
I hope you can see me. Thanks for the introduction, Shashank.
Thank you for joining, and sorry again for waking you up so early. For attendees, it's 7 a.m. in New York right now. I shoved in my talk here to give him a breather. Otherwise, Florian also agreed to talk at 6:30 a.m., which if somebody did to me, I would be really mad. But thanks again. Thanks for joining, and you can share your screen and I'll put it up.
Yeah, thanks for the invitation. I'm trying to share my screen here now. I think you should be able to see it, right?
Yes.
Okay, great. All right, so I'm going to walk you through what we learned here at Mount Sinai in the last 10 months about the antibody response to SARS coronavirus 2, and specifically against the SARS coronavirus 2 spike. So just to start out, you're all familiar now with the virus. We have a model of the virus here on the left side, and there's two major targets of antibody responses that you can see here in this model. One is the nucleoprotein on the inside of the virion, which covers the genome, and the other one is the spike protein on the surface of the virus, which is used by the virus to bind to ACE2 and enter our cells. And so our laboratory started to work on SARS-CoV-2 on January 10th when the sequence came out, and we decided very quickly that we would like to focus on the receptor on the spike protein and the receptor binding domain in terms of understanding antibody responses, because it was known from other coronaviruses that antibodies against the spike can neutralize the virus. And so here on the right side, you just see a crystal structure of the spike protein. It's a trimer. One of the monomers here is shown in blue, and the receptor binding domain of that monomer is shown in red. And so this receptor binding domain is really the business end of the molecule that binds to ACE2. And so early on, we started to express the receptor binding domain and the full-length spike in its trimeric version, and use that as reagents to study antibody responses to the virus. I'm not going through all of that development. That's all published already in spring, but initially this was an academic exercise. We set this up in the lab in February, but in the beginning of March, when the first cases hit New York, we started to talk to our clinical laboratory, and they were very interested in having such a tool in the laboratory to be able to screen potential plasma donors. And so with them, we came up with a setup where we have two-step ELISA, where during the first step, we screen at the set concentration reactivity to the receptor binding domain, and every sample that's above a certain cutoff then gets titered out on a plate that's coated with the full-length spike, which also contains the RBD. And so a sample is only positive if it's positive in both of those assays that are sequentially done. And this setup works very well. It gives us very high specificity, but it's also pretty sensitive. And so we transferred that to our clinical laboratory. They got New York State emergency use authorization already in March, and then FDA emergency use authorization in April. And now there's even a commercial kit that already has a CE mark. What we found initially was that we have 92% sensitivity and 100% specificity. And right now the specificity is still above, way above 99%, and the sensitivity actually improved a little bit. We also shared these reagents early on with more than 250 laboratories worldwide. I think we also shipped a couple of packages to India. and they're available from BI resources, and we also published an open access protocol right away to help people to set up antigen expression and the test setup. Now, what did we do with the assay here? As I said, Mount Sinai initially was very interested in looking, finding plasma donors, people who had already seroconverted to the virus, would then donate plasma, and we could start to treat patients with that plasma. And that was initially really the only thing we had to offer to patients. And by now we have screened more than 70,000 donors by ELISA. And these donors come in two categories. We have PCR confirmed donors who really know that they had the virus infection. We have donors with suspected COVID-19 but without PCR confirmation. I'll show you some data on that in a few minutes. And so far we have treated more than 500 patients with convalescent plasma. There's still some questions about the efficacy of convalescent plasma that are getting solved right now. But it seems that there's some data that suggests if you give it early enough, it does have an effect. So I just told you that initially we had PCR confirmed donors and COVID-19 suspected donors. And that's what you can see here. This is from an initial analysis of about 1,300 individuals. And for the PCR confirmed donors, we found that 99.5% of them actually make antibodies. We only had 3 individuals in this group of 568 people who didn't make antibody. With the suspected donors, it was only about 40% of people who made antibodies, and this was early on, and we still had flu cases here in the city, so we think that a lot of these people in the red here actually never had a SARS-CoV-2 infection, but had other infections or might not have had an infection at all. And then we went on, and as I said, more than 70,000 samples have now been screened. And what I'm showing you here is the result of these screenings. The clinical lab, the way it's set up, they basically specify different titers, low titers, which are 1 to 80 and 1 to 160, intermediate titers, which are 1 to 320, and then high titers, which are 1,960, or 1 to 2,880 or above. So they have a cap on how far they titer out. And if we look now at the people who screened positive, and those were 30,000, we see that very few individuals, about 7%, have low titers, about 20% have intermediate titers, and about 70% of individuals have actually high titers, indicating that people get a robust antibody response. There's a lot of mild and asymptomatic cases in this analysis. They also can develop high and relatively high titers. And what I'm showing you here on the right side is just individuals screened per day over the last couple of months. And the blue line here indicates for each day how many, the percentage of people that had a titer of 1 to 320 or higher. And you can see that it's consistently around 90%. So even over time, we see that, that we get robust titers. Now, the next question that we had was, okay, we can measure antibodies that bind, but do they do anything? Are they functional? And so we perform neutralization assays here, which are based on authentic SARS-CoV-2. These are quantitative assays. And so we took 120 ELISA samples from the ELISA runs and also performed neutralization assays on them. And on the y-axis here, you can see the neutralization ID 50, and on the x-axis, you can see the ELISA endpoint titer, and you can see that there's a very nice correlation. The higher you go, the better the neutralizing activity is, and basically starting at 1 to 320, almost everybody has neutralizing activity in their serum. Now, and if you're interested in the setup of that assay, we also published that open access in detail as a protocol. Now, if you remember, earlier this year, there was a lot of excitement and a lot of reporting about antibody titers that wane relatively quickly. We heard that a lot from the media, still coming back. I hear that all the time. I talk to somebody at the media and on the street, and this notion was based on this paper that came out in Nature Medicine. And what they showed here, they looked at asymptomatic cases and symptomatic cases, and they looked for antibodies to nuclear protein and one spike epitope in a non-quantitative assay, and they saw that over time, over the time period of 8 weeks, about 40% of the asymptomatics lost their nuclear protein titer. While for the symptomatics, it was a little bit better, but did go down too. So we decided we wanted to look at that as well, because of course it's a very important question, how long do these antibodies last? And if they last only 8 weeks, that would be A, problematic, and B, would be really hard to explain with basic B-cell biology. And so we did a similar analysis. We recruited 121 of our plasma donors, and we bled them around day 30, and we asked them to come back around day 82, and then around again around day 148. So that's 5 months. And so what we see is, I'm showing you here ELISA titers. These are GMDs. We see that there is a drop from day 30 to day 82. It's significant, but not very high, or the drop is not very high. And we get another drop from day 82 to 148, but the antibodies don't disappear. And so this is not a very drastic drop. We also analyzed that per initial titer. So what I'm showing you here are stratified in terms of the day 30 titers, and for the high titers, we see, yes, there is a decline, but again, the decline is not very drastic. If you look at the lower titers, it looks a little bit different. We get an initial increase, actually, if you have intermediate or low titers from day 30 to day 82, so the antibody response seems to be a little bit slower. And then we basically have some decline, but again, it's not a very drastic decline. So it seems that these antibodies behave like antibodies should behave after an infection with a virus. I mentioned this graph from this Nature Medicine paper here on the left side, but there was another panel in this Nature Medicine paper in the same figure, actually, that was not picked up by the media, and that showed that in both asymptomatic cases and symptomatic cases, the neutralizing antibody titers are actually pretty stable. And that goes along very well with our data, because we measure antibodies against the spike, and those are the neutralizing antibodies. So I think what I'm showing you here is that very often it's very important how you communicate results, because the negative results will be amplified by the media, while any positive results are very often not of much interest. But we also wanted to know how neutralization in our case looks over time, and that's what I'm showing you here on the left. Again, we have our 121 individuals, and we measured neutralization titers over time, and you can see that there's actually not much difference. There is a decline in GMDs from 176 to 52, but we don't see neutralizing activity disappear over the course of 5 months. And if we do our correlation analysis again, at the 5 months time point, there's still a nice correlation between ELISA titers and neutralizing activity, indicating that there was no major shift in epitopes, for example. Now, what does this all mean, right? So we find antibodies, we find neutralizing antibodies, but you know, what does that mean for us? What does that mean for immunity? And so we're very interested in correlates of protection. And I'm showing here 2 papers from Dan Baruch's group, work that was done in non-human primates and published earlier this year. And on the left, you see an experiment where non-human primates, rhesus macaques, were challenged with SARS-CoV-2. And you see here the RNA copies per mil. I think this is BALF. And you see the animals get infected, and then over time they clear the virus. And if they then re-challenged, it's very hard to infect them. There's a blip here for 2 animals, but they're basically protected from reinfection. The same group did another experiment where they vaccinated non-human primates with different DNA vaccine constructs, not really a vaccine that was supposed to be developed as human vaccine, but they just wanted to test how a vaccine would fare. And because they had different constructs, they got different titers of neutralizing activity. And that allowed them to do correlation analysis between the neutralizing activity and the protection. And what they see is a very strong correlation between neutralizing activity and protection. The more neutralizing antibodies you have, the better these animals are protected. And if you look at the titers here, they're actually not that high. We're talking about 1 to 100 titers. What is of note here is also that they looked at CD8 T cells, and CD8 T cells, which are also induced by DNA vaccination, were not a correlate of protection in this model. Now, we have a lot of correlates of protection for humans for different vaccines, and they're very important because basically, if you have a good correlate, it can tell you if somebody is susceptible or if somebody is not susceptible to infection. We have this, for example, for hepatitis A, hepatitis B, flu, the well-known 1 to 40 HI titer, which reduces your risk of getting influenza by 50%. And also measles, for example, if you have a microneutralization titer of 1 to 120, you're likely going to be protected. So it would be very important to have something similar for SARS-CoV-2 that would allow you to tell a person if the person is protected, either by a vaccine or by a natural infection, or if that person might need to get a booster dose of a vaccine in the future, right? And so neutralizing antibodies are a good candidate here. And as you can see, in many cases where we have correlates of protection, it's really antibodies that are the correlate. It doesn't mean that T cells are not important. Probably just means that T cells are much harder to analyze and to measure. And so we got a first glimpse of neutralizing antibodies being a potential correlate of protection. I don't know if you have seen this story. It's a very nice paper by the Krammer group, and there was also a story about it in the New York Times. Basically, a commercial fishing vessel, the whole crew was recruited and enrolled in a study, and their neutralizing antibodies were measured. And of the 122 people on that boat, only 3 had neutralizing antibodies to SARS-CoV-2. And then the ship left, and as we know from cruise ships, if you're on a ship, very close quarters, there's a lot of interaction. If you have an outbreak with SARS-CoV-2, a lot of people get infected, and this is what happened on this boat. They had an outbreak with an attack rate of about 83%. The 3 people who had neutralizing antibodies did not get infected. And while this is a very small number, it's statistically significant. And I'm sure you have followed the news yesterday about Pfizer. They see effectiveness or efficacy now with their vaccine, and their vaccine is really inducing neutralizing antibodies, not much else. So I think this bodes very well for neutralizing antibodies as correlate of protection and for the vaccines that are in development. But we wanted to look at that ourselves. Actually, before that study came out in April already, we were discussing how to look at that. And together with Viviana Simon, a colleague of mine here at the department, we started a study that's called PARIS, or Protection Associated with Rapid Immunity to SARS-CoV-2. And the aim here was to see if reinfections can happen. We know now they can happen, but they seem to be very rare, and to determine protective titer. And so we set this up in a way that we have 400 participants, and 50% of the participants that are enrolled already have antibodies from prior infection, and another 50% are antibody negative. And then we follow them. We bleed them every 2 weeks. We take saliva samples, which allow us to go back and see if somebody was infected. And we also, if somebody becomes symptomatic, that person has to come in and is tested for SARS-CoV-2, and is also tested with a BioFire respiratory panel for infection with other respiratory viruses. We started that on April 27th. We are almost fully enrolled. I think we're 380 or 370 individuals right now. But as you know, New York has had over the summer a very low attack rate. It's now creeping up again. So I don't have any data to show you about what the protective titer is. Few months. What I can show you is antibody levels over time. And so these are the positives in the Paris study. What I want to point out here is that 2 things. First, day 0 here is the enrollment day. many of these people were infected months before they were enrolled. A lot of the people that we initially recruited in spring had their infection, back in March. The other thing that I wanted to point out here, this is area under the curve, and we basically don't end with an 1 to 2880 endpoint that we titer out completely, which gives us a much better idea of fluctuations and how high antibody titers actually go. And so what you can see here is titers of these individuals over time. And what we see is in some cases we have an initial decline. This might still be coming down from the original peak after infection. And then these titers stabilize. There's not much going on. You see fluctuation, but that could also be variation in measurements, day to day. But we really see that these titers are fairly stable and they seem to hit the baseline. We see similar things now with the other study that I just showed you, where we now also have some data from a month 7 time point, and we also don't see any declines anymore between months 5 and month 7. So it looks like we hit a baseline that's maintained by long-lived plasma cells in the bone marrow, and I don't think that these antibodies will just go away. And because we only had 400 people here at Mount Sinai, and because we had low attack rates, we now expanded this study to 9 sites across the US with a target enrollment of about 12,000 individuals. So I hope we'll get data about the correlate of protection within the next few months. There's a lot of infections in the US right now, which of course helps to generate data, although it's sad. And the last point that I wanted to touch is an ongoing sero survey that we are doing in New York City. This has been published last week in Nature. We have initiated that in the beginning of February, way before we had the first cases. We were thinking that we should start something to look at seroprevalence to the virus in the city. And so we started to collect residual samples from our clinical laboratory. And so we established 2 groups here for this design. We had what we call an urgent care group. So those are samples that come from people who come to the emergency department and from people who are then admitted to the hospital. And we test a few hundred of those per week. And the rationale behind this group was that if we have a major outbreak, a major epidemic in New York with SARS-CoV-2, a lot of people with SARS-CoV-2 will come to the hospital, and we should see a peak there, right? There should be a strong increase. So this was basically our positive control group. And then we had a second group that we call routine care group. And so those are people who have to come to the hospital no matter what. This is OBGYN, basically labor and delivery, oncology patients, surgery and pre-operation assessment, and cardiology. So these people don't come to the hospital because they have SARS-CoV-2. They come to the hospital for other reasons, and they even come during an epidemic. And we had 250 to 500 samples from that group per week. The urgent care group is skewed towards older males. The routine care group is closer to the population of New York City, but we have a skew towards females because of the labor and delivery group, and we are underrepresented in children in this group. But other than that, it's relatively close to the population. Just to remind you of how the pandemic, or the first wave of the pandemic, looked in New York. We had our first confirmed case on March 1st, so that was detected at Mount Sinai, I think on the 28th or 29th of February, but then confirmed on March 1st. On March 7th, the state of emergency was declared, and then on March 11th, we had capacity for the first time for more than 500 PCR tests. So you can imagine that, I'm showing here in green the case numbers. These are the official case numbers. There were many, many, many more cases here in the beginning, probably a huge curve, but they went undetected because we didn't even have the measures to screen for that. And on March 22nd, we got a shelter-in-place order, and basically the city locked down, and relatively shortly after that, 2 weeks after that, the cases peaked and then went down. and by May 24th, we started to hit what became a baseline over the summer. And if you look at the deaths basically follow that with some kind of delay. These are official deaths where SARS-CoV-2 was confirmed. There's additional deaths where SARS-CoV-2 was suspected to be the cause of death. I'm not showing that data here. That's approximately an additional 20 to 30%. Okay, so let's look at this urgent care group first. What you can see here on the right side is the seroprevalence in the urgent care group starting by the week of February 9th. And then I'm showing you data weekly. So we have a little bit of seroprevalence already in the end of February, in the beginning of March. There was a blip there. There were certainly some samples that were positive. But then we really get a strong increase around March 22nd, that's when it starts. And then we get a peak around April 19th, which is, basically right after the peak of cases here. And we peak at 60% or a little bit more than 60% seropositives. And this is, of course, when all the patients came in with SARS-CoV-2 into the hospital. And then after that, the seroprevalence in this group declines, and it goes down to between 20 and 22%, and it stays there. And what happened here is around May 24th, the wave was over, and after that, what we see is people who come to the hospital or to the emergency department for other reasons, maybe because they broke their leg or because they had a heart attack. But not necessarily because of SARS-CoV-2 anymore. So this flat curve here represents more or less what we see now in our regular patients. This urgent care group, the seroprevalence here, which is shown as a black line, overlaps very nicely with the COVID-19 diagnosis in that group. So that also confirms that we actually pick up the wave, and then we also titer these individuals, so we get quantitative titers, and what you can see here is that initially there are some individuals with lower titers, but then we get this huge increase in titers, and after the wave the titers go down a little bit, and we know now that severe cases usually have higher titers, but then afterwards the titers stay basically stable. If we now look at the routine care group, again, this represents the regular population much better. We also see some very low seroprevalence already in late February, and then we really start to see an increase in seroprevalence around April 5th. It climbs up, and then it hits about 20%, and it stays there. And it stays there until July. That's kind of when the data ends here. But it shows you that the seroprevalence is pretty much stable, so it doesn't decline. And this number of 20% here is very, very similar to the number in the emergency, in the urgent care group after May 24th. So we end up with approximately the same number. And if you look at titers here, we have some higher titers initially already here in the beginning of the study, and then it climbs up, and the titers stay steady over time. Again, this kind of suggests that the antibody titers are relatively stable over time, because the people we titered out here on the right side, for example, have most likely been infected in early spring with the virus. And if we look at the routine care group and the official cases, what you can see here on the left side is the number of official cases in red, and then the seroprevalence in the routine care group, and you see that the seroprevalence really follows the peak of officially infected individuals. We can also do the same with titer, and you see that the titers are climbing up, and then they're staying the same. And we can also look at cumulative cases, which is basically here this dark red line, and then I also have shifted that for 7 days and 14 days and 21 days because seroconversion takes time. And you see that the increase in seroprevalence actually overlaps very nicely with those cumulative case numbers. Of course, Initially, we missed a lot of cases, as I said, so the curves probably look similar if we would know all the cases, but initially we didn't have the tools to actually diagnose a lot of cases. And so that brings me to my conclusions. First, I think we have evidence, and this is also coming from a lot of other labs now, that humans induce a solid antibody response to SARS-CoV-2, even after mild infection. In general, you see higher antibody responses after severe infection, but this is not an absolute rule, and we had mild and asymptomatic cases that also had sky-high neutralizing antibodies. The antibody response looks fairly normal. If you look at B-cell biology, it's basically what's supposed to happen. We see an initial strong increase, which is most likely driven by plasmablasts. They make tons of antibodies, but they're very short-lived. They just die after a few days. And then this antibody sticks around. We have a half-life of about 21 days for IgG. And this is probably why you see the antibody staying up for several weeks. And then at some point you see a decline. That's probably because the initial antibodies start to fade away, and the plasmablasts that produce them are not there anymore. And then it goes down to a plateau or a certain level that is then maintained by long-lived plasma cells. So again, that's exactly what you would expect from a normal B-cell response. Now the $1 million question is that level where it flattens out above or below a protective threshold. If it's below, a person can likely get reinfected. If it's above, the person is likely protected. But we don't know what that threshold is yet. We also found that antibodies binding to the spike protein correlate very well with neutralization, which is not rocket science. That was expected, but it's nice to see it, and it's nice to see it with solid data. The Berry study that I showed you is ongoing and was extended to other locations across the US, so we hope that we'll know that threshold of protection soon. The takeaways from the Sero survey is that potentially we had low-level seroprevalence in New York City starting already in the week of February 23rd, which would mean people might have been infected already in New York, some people, by the beginning of February, and that goes along very well with phylogenetic analysis. We see that our urgent care control group really worked. We see this humongous peak in cases and seroprevalence that goes up to 60%, and the cases and the seroprevalence increase in seroprevalence overlaps. And if you look into the routine care group, which again basically reflects better what's going on in the population, we see already a seroprevalence of 19% in the mid of April. That then goes up a little bit higher to about 20.3%. And if we now use those numbers to calculate how many people in the city were infected and the infection fatality rate, we find that about 1.7 to 1.8 million of New Yorkers have already been infected. We have a population of 8.4 million. And that the infection fatality rate is relatively high. It's 0.97, so almost 1%. Keep in mind, this is based on the officially confirmed deaths, SARS-CoV-2 deaths. There's additional deaths that were not put into this calculation that are strongly suspected to have died of SARS-CoV-2. So that would be even higher. That's above 1%. And I think the main take-home message from this is that if you have a huge wave of infections and your hospital system, your healthcare system is completely overwhelmed and you cannot take good care of these patients anymore, then your infection fatality rate goes up. And I think this is something that needs to be communicated more, specifically now in Europe and in North America. And with that, I'll just like to acknowledge a lot of people. We had a lot of collaborations here at Mount Sinai from the get-go, but also outside of the US. Initially, when we tried to set up the assay, I did not have positive control samples, and I got them from collaborators in Finland and in Australia. That allowed us to really set everything up in February. And we have, as I said, a lot of support here from the school, from the department, and also from our funding sources at the NIH through civics and CIDRs. And with that, I'll stop, and I'm happy to answer any questions.
Thanks a lot, that was an exhaustive body of work. I'll take questions in reverse order. Why do you think just 20% seroprevalence is able to reduce disease incidences so significantly?
I don't think it does. I think that the control measures reduced disease incidence. Of course, if you have 22% seroprevalence, that might reduce the reproductive number to a certain degree. It might make it easier to control the virus with additional control measures, but that's not the reason why SARS stopped circulating. And actually, right now the cases are going up again, and we're getting into trouble again. So we're not talking about herd immunity here, unfortunately, which is has become a loaded term and probably shouldn't be used in that sense anymore.
Yeah. All right. Second question is between subunit vaccines against spike or whole virus inactivated vaccines, which has other components, which ones you think would be better?
I think what would be good is to have high neutralizing antibody titers, and we get those right now with the RNA vaccines, but even more so with the recombinant protein vaccines. Novavax has really nice data, and I think Sanofi Pasteur will also come out with really nice data. But I think also the inactivated vaccines give significant amounts of neutralizing antibodies. I don't think that antibodies against the E or the M or the NP will really contribute to protection. But of course, making an inactivated vaccine is a way to make a vaccine. And right now we need all the vaccine production capacity that we have to get this under control.
Okay, one question is based on the seropositivity, can it be traced back when person was exposed to the infection?
That's hard. You could hypothetically, but not every person has the same kinetics, right? But hypothetically, you could try to do that when they're still on the decline. I think once they hit the plateau, it's not possible anymore, even if you get 2 or 3 time points, because you would only see kind of a flat line, and that would really make it difficult to do so.
All right. Next question is, instead of neutralizing antibody titers, wouldn't B cell memory be a better correlate for protection?
So I think memory B cells and also T cells are playing a big role in controlling this virus in terms of reinfection, because you have a relatively long incubation time, right? So these cells have a lot of time to get reactivated and fight back. And so even people who might not have antibodies anymore, if they have a memory B cells that recognize the virus, they might still be able to avoid getting sick again. Now, the problem is, cellular correlates of protection are usually not something that you can easily establish and easily measure. If you wait for a year or so and you look for memory B cells, it's really hard to find them, right? You have to kind of label them, you have to sort for them. Those are not assays that can really be used in clinical practice or not even in large phase 3 trials. Imagine doing FACS analysis in 45,000 people at several time points. So these things might correlate with protection, but they're really hard to measure in reality, right?
Yeah. All right. One point from Annapurna. In London longitudinal study, it has come out that higher titers of neutralizing antibodies, they actually correlate with disease severity, which is contrast to what's been observed in New York. Is that correct?
No, we also see higher titers in people who have more severe disease. That is known, but it's not a perfect correlation. As I said, there are also asymptomatic people who make sky-high antibody titers. The highest antibody titer I have seen so far was an asymptomatically infected child in Seattle with neutralizing antibody titers, I think in the 10,000s. But in general, the trend is there. People who are more severely sick make stronger antibody responses, at least initially.
Mm-hmm. Okay. One question then I'll ask regarding cross-reactive antibodies between seasonal and current pandemic virus. What implications it will have either ways? Seasonal antibodies for SARS, like seasonal viruses, will they have any implication on current virus? Current virus antibodies, will they have any implication on seasonal viruses? How it's going to play out?
Yeah, I don't have the answer for that, but it's super interesting. So before SARS-CoV-2 infection, we see very little cross-reactivity. But what you see is an original antigenic sin effect. Once somebody gets infected with SARS-CoV-2, they get increases in titers to HKU1 spike and to the OC43 spike, which are beta coronas, but not to 229E or an NL63 spikes, which are alpha coronas. So there is a back boost, and it's not targeting the RBD, it's targeting probably the S2. We clearly see that there's a preprint out together with Adolfo's group, Adolfo Garcia Sastra's group, and other groups see that now too. So if that has an impact on HKU1 and OC43 circulation is not clear. It could have, and it might basically maybe not eliminate, but suppress these viruses to a certain extent. But it's all speculation right now. This is something that I'm personally very interested in.
What about other way around? The antibodies which will be generated due to exposure to pandemic virus, will they have any effect on seasonal virus infections? Will they replace some of the seasonal viruses eventually?
No, that could be. I mean, we have seen this for flu, right? For example, H3 and 2 eliminated H2N2 circulation. The pandemic H1N1 eliminated the seasonal H1N1 and so on and so forth. So it's really hard to say what's going to happen, but one possible outcome could be that there's less OC43 and less HKU1 circulating in the future. So again, it's going to be interesting to look at that.
All right, with that, I think we are done with the questions. Thank you again for joining so early in the morning and giving this very nice talk.